Hello everyone and welcome to today's session. And today we'll be discussing the NCERT practice questions for the chapter Molecular Basis of Inheritance. Before starting the lecture, just want to give you one information that the images, text and other materials used in our presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So, what are the objectives of today's session? So, today we will understand the basic concepts related to the chapter Molecular Basis of Inheritance by discussing the NCERT practice questions, which are related to this chapter. Okay, so today we'll be discussing these 27 questions which are related to chapter Molecular Basis of Inheritance. Let's begin with the first question. So what is the first question here? You have to tell the name of the enzyme that catalyzes the process of DNA replication. Okay. So DNA replication is also known as DNA duplication. More copies of DNA are synthesized. Now let's have a look at the option. Option number A is DNA ligase, B is DNA polymerase, C is RNA polymerase, and D is helicase. So what is the correct answer? Okay, so let's have a look at the answer. So answer is DNA polymerase. This is the correct answer. Option number B is the correct answer. Okay. Now this is a table showing you DNA about information regarding DNA polymerase and RNA polymerase. See, DNA polymerase, this is the main enzyme responsible for synthesizing the new DNA strands during the replication. And in which direction it polymerizes the DNA? It polymerizes the new DNA in 5' to 3' direction. Means the new strand which is being synthesized, okay, and this strand is complementary to the template strand. This strand is being synthesized in the 5' to 3' direction. If you talk about RNA polymerase, this is involved in transcription process. Okay, in transcription, what is being formed? From DNA, RNA is formed. So RNA polymerase is DNA dependent. Okay, clear? Here in this image, you can see this is the DNA polymerase. This is using this template DNA to synthesize the complementary strand. Clear? It synthesizes in 5' to 3' direction. That's why on lagging strands, Okazaki fragments are produced. Okay, so let's clear. DNA ligase was one of the options. So DNA ligase joins the Okazaki fragments which are produced on the lagging strands. Okay, so it makes phosphodiester bonds between the DNA fragments. Then helicase it unwinds the DNA double helix by breaking the hydrogen bonds between the nitrogenous base pair. Okay, this unwinding is very important. Okay, so DNA is double stranded. It needs to be unwind. After unwinding, it becomes single stranded, and then the single stranded DNA can be used as a template DNA. Okay, so you can see here, you can see this helicase which is joining the Okazaki, you can say unwinding the DNA, and this ligase, this is involved in joining the Okazaki fragments together. The next question is genetic code is it universal? Is it overlapping? Is it ambiguous, or it is non-degenerate? What is the correct answer? So let's have a look at the answer. See, genetic code is universal. This is the correct answer. Okay. Now, what do you mean by it is universal? This genetic code is nearly identical in all the different species, from bacteria to humans. So if you have the codon AUG, it will code for methionine in all the organisms. So same codon is coding for the same amino acid across the different species. Okay, so we have initiation codon, which is AUG, which is coding for methionine, and the stop codon, they don't code for any amino acids. Other options are incorrect here because genetic code is not overlapping. Each codon is read independently. Now, genetic code is non-ambiguous, means each codon is specifically coding for one amino acid only. Now, genetic code is non-degenerate. Okay, means multiple codons can code for the same amino acid. Means degeneracy exists. That's why we have 64 codons, but the amino acids coded by the 64 codons are only 20. Because more than one codon is coding for the same amino acid. Now, next question, the number of base pair in human genome is approximately. What are the options? 3.1 billion, 3.1 million, 31 million or 31 billion? What is the correct answer? Okay, so what would be the correct answer from this option A, B, C, D? So let's have a look at the answer. See option number A, 3.1 billion. This is the right answer. Okay. So Human Genome Project reveals the size of the human genome. According to Human Genome Project, human genome has approximately 3.1 billion base pairs. The next question, Messelson and Starr experiment. It proved what? Okay, what are the options? DNA replication is conservative. Option number B, DNA replication is dispersive. Option number C, DNA replication is semi-conservative. And option number D, it proved that mutation occurs in the DNA. What is the correct answer here? So let's have a look at the answer. See, this is a very straightforward question here. Option number C is the correct answer. DNA replication is semi-conservative. Means whenever new molecule of DNA is synthesized, one will be the parental strand, another one will be the newly synthesized strand. Okay, so Messelson and Starr, they did experiment in 1958 to prove that DNA or DNA replication is semi-conservative. So what they did initially, they grew E. coli bacteria in a medium containing heavy nitrogen, means N15, for several generations to label the DNA. So DNA is made up of nitrogenous bases, so nitrogen will be labeled. Then what they did, they transferred the bacteria to a medium with light nitrogen, means N14, and allowed the E. coli to replicate. So initially, when the E. coli were grown in this heavy nitrogen, both the DNA strands were labeled with N15. So after centrifugation, they got only one heavy band. Then they transfer this E. coli bacteria into a medium containing N14. Then after first generation, what did they observe? That only hybrid band was obtained. So this was the intermediate band, and here one strand had N14, another strand has N15. Then again after next generation, after 40 minutes, means generation 2, they got two bands, light band and hybrid band, and there was no heavy band. So this proved that DNA replication is semi-conservative. So this light band had, in light band, both the DNA strands will have N14 and N14. Clear? Now let's move on. Okay, so after one generation, they got a band with intermediate density. Okay, Means one strand had N15, another strand had N14. Okay, then after second generation, okay, after 2 generations, they got 2 bands, light density band and intermediate density band. So this proved that DNA replication is semi-conservative. And one of the strand, the original strand, act as a template, okay, for the synthesis of new strand which is complementary to that strand. Next question, lac-operon is an example of what? Options are positive regulation, negative regulation, both A and B, and neither A nor B. What is the correct answer? Okay, so let's have a look at the answer. Answer here is option number C, both A and B. Okay, so lac-operon exhibit both positive and negative regulation. Let's talk about the negative regulation. So, what happens in the negative regulation? The repressor protein, this will inhibit the transcription by binding to the operator region. Okay, when repressor is, you can say, binds to the operator region, then the RNA polymerase cannot bind to the promoter region. So this prevents the transcription. So when the inducer, which is lactose, is absent, transcription is prevented. So this is known as negative regulation, which is done by the repressor protein. Okay, so this is negative control of the lac-operon. As the repressor, this is blocking the or inhibiting the expression of the genes. Let's talk about positive regulation. So there is one catabolite activator protein. So this protein, when bound to cyclic AMP, acts as a positive regulator. Okay, when the glucose conditions are low, okay, now we are talking about glucose, not lactose. So now in the conditions when glucose are low, this cap cyclic AMP complex binds and stimulates the transcription. So that's why this is known as the positive regulation. Okay, so this is positive control as it actively promotes the gene expression. Okay, this catabolite activator protein, this is basically activating the gene expression. The next question, this is again very straightforward question. Number of codons in genetic code is? What are the options? 16, 32, 64 or 128? Again, this is a very straightforward question here. So let's quickly have a look at the answer. See, answer is 64. Option number C is the correct answer. Okay, so how do you have 64 codons? And what is codon? So codon is basically triplet of nucleotides which is present in the messenger RNA. This codon codes for a particular amino acids. So basically you have 4 nitrogenous bases, okay, in RNA. So you have adenine, uracil, guanine and cytosine. Now how can you calculate 64 codons? See, there are 4 possible nucleotides at each position. Okay, but the codon is made up of only 3 nucleotides. But we have 4 possible nucleotides which can be present at each of the position. Okay, so codon has 3 positions. So this means 4 by raised to power 3, that will be 64. So these are the total possible combinations of the codon. Okay, so this 64 codon codes for 20 amino acids. Now next question, again a very straightforward question. Initiator codon is? What is the sequence of the initiator codon? So options are UAG, AUG, UGA and UAA. What is the correct answer? Okay, so let's have a look at the answer. Answer is AUG, which is option number B. Okay, so AUG codes for methionine and serves as a start codon for the protein synthesis. UAG is a stop codon known as amber. UG is a stop codon known as opal. And UA is a stop codon known as the ochre. They don't code for any amino acids. Now, next question is related to Griffith experiment. So, Griffith experiment demonstrated what? What are the options? DNA is a genetic material, transformation, transaction, of a translation. What is the correct answer? Okay, so let's have a look at the answer. See, option number B, transformation, is the correct answer. Griffith experiment demonstrated transformation. So he said there is some transforming principle, okay, and something is being transferred from the S strain to the R strain. And the R strain bacteria, they were transformed into S strain bacteria, which are virulent. Okay, so Griffith showed that non-virulent bacteria, means the R strain bacteria, could transform into virulent, means S strain bacteria, by some transforming principle. This transforming principle was later identified as the DNA. Okay, DNA as a genetic material was proved by the experiment done by Avery McCarty. Okay, so Avery et al., they proved that DNA was a genetic material. Okay, not Griffith. Griffith told about the transformation. Now there was one term, transduction. So transduction, this involves virus-mediated DNA transfer. So when virus infects the host cell, virus transfers its nucleic acid into the host cell, and the protein coat, it remains outside. Translation, we know that it is synthesis of proteins from the messenger RNA. So this was the experiment done by Griffith. Okay, so what he did, he injected the mice with living S strain bacteria. In this case, the mice died. In another case, he injected the mice with living R strain bacteria. In this case, the mouse survived. Now he injected the mice with the heat-killed S strain bacteria. Okay, in this case, the mice survived. And in the last setup, what he did, he injected the mice with a mixture of heat-killed S strain bacteria and R strain bacteria. So usually, what should happen? The mice should survive. But in this case, mice died, and from the dead mice, S strain bacteria were isolated. Okay, so from this experiment, he proved that the R strain bacteria were transformed into S strain bacteria by a transforming principle. So something was being transformed from this S strain bacteria to R strain bacteria. Now, next question, DNA fingerprinting is based on what? Protein sequences, repeated DNA sequences, single copy genes of a RNA sequences. What is the correct answer? Okay, let's have a look at the answer. See, DNA sequences is based on repeated DNA sequences. Okay, so some sequences, they are repeated. They are known as repetitive DNA sequences. Okay, like satellite DNA. Satellite DNA is a kind of repetitive DNA. Okay, so DNA fingerprint is based on repetitive DNA sequences, and more specifically, you can say VNTRs, variable number of tandem repeats. Now, what is this VNTR, or variable number of tandem repeat? So see, some of these sequences, Okay, these sequences are repeated multiple times, and these sequences are adjacent to each other. So tandem means adjacent to each other. Okay, now the copy number of this VNTR is different in different organisms. It is unique. So DNA fingerprint is based on repeated DNA sequences, specifically focuses on regions called variable number of tandem repeats and short tandem repeats as well. See, variations in this repetitive DNA sequence are unique to the individuals. Okay, why protein sequence is not the basis of DNA fingerprinting? Because they are too conserved and they are not unique enough. Okay, we require variations. We do not require conserved region. Single copy genes, they are also not variable enough between the individuals. RNA sequences, this is also not stable enough, and it can vary with cell type and the conditions. So that's why the correct answer is repeated DNA sequences. The next question, the enzyme RNA polymerase. Options: requires a primer, does not require a primer, requires DNA ligase, and requires reverse transcriptase. What is the correct answer? See, let's have a look at the correct answer here. See, option number B, does not require a primer. This is the correct answer. Okay, so RNA polymerase does not require a primer. DNA polymerase requires a primer. Okay, what are the characteristics of RNA polymerase? So RNA polymerase can initiate RNA synthesis de novo. What do you mean by de novo? De novo means from the scratch, and it doesn't require any primer. Okay, RNA polymerase does not require a primer to initiate the RNA synthesis. This is one of the key distinguished features of RNA polymerase compared to DNA polymerase. Okay. So DNA polymerase requires the primers, not RNA polymerase. Requires DNA ligase. This option is also incorrect because ligase join the DNA fragments. And one of the options would requires reverse transcription. This is also incorrect because reverse transcription is used to make DNA from RNA. See, next question is the sugar present in RNA is. Okay, so this sugar is a pentose sugar made up of 5 carbon atoms. So this sugar present in RNA is ribose sugar, deoxyribose, glucose or fructose. Again, a very straightforward question. So RNA is ribose nucleic acid. So what should be the name of the sugar? It is ribose, which is option number A. Okay, so RNA contains ribose sugar while DNA contains deoxyribose sugar. One of the options was glucose. Glucose is a hexose sugar, means it consists of 6 carbon. It is not found in nucleic acid. Then fructose is also a hexose sugar. It is not found in nucleic acid. Here, this is DNA and RNA. So RNA consists of ribose and DNA consists of deoxyribose sugar. Okay, that's why DNA is more stable also, because in deoxyribose this 2' OH is absent. The next question, Hershey and Chase experiment used. Option C14 and N15, P32, S35, H3, C14 and N15 and S35. What is the correct answer? Okay, so let's have a look at the answer. The answer is option number B, P32 and S35. Okay, Hershey and Chase experiment was a landmark study that proved DNA is the genetic material, not proteins. So this experiment, radioactive isotopes were used. Okay, so P32, which is phosphorus 32, S35 is sulfur 35. So P32 will label the DNA because DNA consists of the phosphate group. Okay, DNA backbone is made up of sugar and phosphate. Okay. Now S35 will label the proteins because proteins they made up of amino acids and there are some sulfur containing amino acid also like cysteine and methionine. Next question, nucleosome consists of. Again a very easy question here. So nucleosome is present in the eukaryotic cell. They help in chromosome DNA packaging. What are the options? DNA only, histones only, DNA wrapped around histones, or RNA and proteins? What is the correct answer? Okay, so let's have a look at the answer. So option number C is the correct answer. So what is nucleosome? Nucleosome is a, you can say, unit. Okay, it is a structural unit of the chromatin. And what a nucleosome is made up of? See, the negative charge DNA is wrapped around the positively charged histone octamer. This is known as a nucleosome. Each nucleosome carries around 200 base pairs. Okay, so when DNA is wrapped around these histone octamer, this is known as a nucleosome. Okay, so it is the basic structural unit of the chromatin. So nucleosomes, they combine to form the chromatin. Okay, now DNA alone cannot form the characteristic nucleosome structure. So, okay, so along with DNA, histone proteins are also involved. So DNA is wrapped around this histone octamer. Clear? So this nucleosome contains DNA with histones, not RNA. Here you can see this is a nucleosome. This is DNA. DNA is wrapped around the histone octamer. This green color is histone octamer. Clear? This is the structural unit of the chromatin. This helps in DNA packaging. Now next question, the number of hydrogen bond between G and C base pair. Again, a very straightforward question. Number of hydrogen bond between G and C. Guanine and cytosine is 1, 2, 3 or 4. So let's have a look at the answer. The answer is option number C, 3. Okay, now A, pairs with T by 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. In this image, you can see these are the 3 hydrogen bonds between G and C, and there are 2 hydrogen bonds between A and C. Now next, during transcription, RNA is synthesized in option 5' to 3' direction, 3' to 5' direction, both direction of a random direction. What is the correct answer? Okay, so let's have a look at the answer. See, option number A is the correct answer, 5' to 3' direction. Okay, so RNA polymerase will always synthesize the mRNA in the 5' to 3' direction. This is the fundamental aspect of RNA synthesis that occur in all living organisms. So here you can see, this is image showing you direction of the synthesis of the RNA. RNA is being synthesized in 5' to 3' direction. And this is the template DNA. Okay, and complementary to this, RNA is being synthesized. Clear? Next question. The main application of DNA fingerprinting is in. Okay, you have to tell about the applications of the DNA fingerprinting. So options are gene therapy, forensics, and genetic engineering offer mutation studies. What is the correct answer? So let's have a look at the answer. C, option B, forensic science is the correct answer. DNA fingerprinting is also known as DNA profiling or DNA typing, and it is primarily used in the forensic science. Okay, so it is used for crime scene investigations. It is used for identify suspects or victims. It is used for paternity testing, whether who is the mother and the father, who are the parents. It is also involved in missing person cases as well, to identify missing person. So it is also involved in establishing the biological relationship. Means who is the parents, who is the father, if this missing person is related to that person. Next, Human Genome Project was completed in which year? So you have these four options: 2000, 2001, 2003, and 2005. What is the correct answer? See, option number C is the correct answer. 2003 is the correct answer. So Human Genome Project was a 13-year project completed in 2003. Now, what is the timeline of Human Genome Project? It was launched in 1990. The working draft was announced in 2000. Okay, the project was declared complete in April 2003. Now, the total number of genes in humans is approximately. Okay, options are 100,000 genes, 50,000 genes, 20,000, 25,000, or option number D is 10,000 genes. What is the correct answer? Okay, so answer is option number C. 20,000 to 25,000. This is the correct answer. Okay, so human genome consists of approximately 20,000, 25,000 protein coding genes. Okay, so less than 2% of the genome codes for the protein, 98% is the junk DNA. It's mostly made up of repetitive DNA. So earlier the number of genes which were estimated was you can say very high, but after Human Genome Project it was you can say revealed that the human genome consists of around 20,000 to 25,000 protein coding genes. Next question, the percentage of human genome that codes for the protein is. So options are less than 2%, about 10%, about 25%, or more than 50%. What is the correct answer? So let's have a look at the answer. See, less than 2%, this is the correct answer. Okay. So about 1 to 2% of the human genome consists of protein coding sequences, which are exons. Exons are the coding regions. So this corresponds to approximately 20,000 to 25,000 protein coding genes. So less than 2% of the human genome codes for proteins. Okay, 98% is the junk DNA. They don't code for any proteins. Next one, see the chromosome with maximum number of genes is. What are the options? Chromosome 1, X, chromosome Y, or chromosome 21? What is the correct answer? The correct answer is chromosome 1. Option number A is the correct answer. See, chromosome 1 has maximum number of genes. It is the largest human chromosome. It contains approximately 249 million base pairs and represents about 8% of the total DNA in the human cells. Now, approximately 2,968 genes are present on the chromosome 1. Now, VNTR stands for what? Again, a very straightforward question. Options are variable number of tandem repeats, variable nuclear tandem repeats, variant number terminal repeats, or variable number terminal repeats regions. What is the correct answer? Okay, so we have option number A as the correct answer, variable number of tandem repeats. Okay, so these are basically repetitive DNA sequences. Okay, they are tandemly repeated, means they are adjacent to each other. Okay, these sequences are adjacent to each other. So there is one sequence that is being repeated multiple times. Okay, but the copy number is different. In some, or in one individual there are 4 copies, in another individual there can be 6 copies. Okay, so VNTRs are basically variable in number of repeat units among different individuals. Means in one individual there can be 4 copies, in another individual there can be 6 copies of that sequence. See, GATA, this is being repeated multiple times. Okay, in one individual there are 4 repeats, another individual there are 6 repeats. Next question, the technique of DNA fingerprinting was developed by whom? Options: Francis Crick, James Watson, Alec Jeffrey, and Frederick Sanger. What is the correct answer? See, option number C, Alec Jeffrey is the correct answer. So Alec Jeffrey developed DNA fingerprinting using VNTR as the probes. Okay, so he radio labeled VNTRs and he used these radioactive VNTRs as probes. What was the contribution of Crick, Watson, and this Francis Crick and James Watson? They discovered the double helix structure of the DNA. Sanger, he developed the Sanger sequencing method. So by Sanger method, we can sequence the DNA. So this is an image showing you Sir Alec Jeffrey, who discovered the DNA fingerprinting. The next one, the percentage of DNA sequence similarity between any 2 humans is. Okay. How much genome is similar? How much DNA sequences are similar in 2 different individuals of humans? So options are 90%, 95%, 99.9%, or 100%. You have to tell about percentage of DNA sequence similar between 2 humans. Option number C is 99.9% DNA sequence is similar between 2 humans. Only 0.1% variation occurs. Okay, or only variation occurs in only 0.1% of the DNA sequence. Okay, now this smaller 0.1% difference account for all the genetic variations among the humans. Next question, SNPs in human genome refers to. Okay. Options are single base differences, multiple base differences, gene deletions, or chromosome variations. What is the correct answer? So let's have a look at the answer. Option number A is the correct answer. Single base differences is the correct answer. So SNP is basically single nucleotide polymorphism. there is a difference in the base at only one position. Okay, it is single base pair variation in DNA sequences that occur at specific position in the genome. So example is sickle cell anemia. It is the most common type of genetic variation among people. Now these variations occur approximately once in every 300 nucleotides. So it is so common, you can see. So here you can see this is an image showing you single nucleotide polymorphism. It is causing a change in the single base pair, or you can say single base. So these are three individuals. You can see rest of the sequence is same except at this fourth position. So this is an SNP. In one individual you have A, another you have G, in third individual you have T. So these are polymorphism, these are variations, but at only single point, at only single position. Only one base difference is there between different individuals. Next question, see BAC stands for what? Options are basic artificial chromosome, bacterial artificial chromosome, bacterial accessory chromosomes, basic accessory chromosomes. What is the correct answer? Okay, so let's have a look at the answer. See, the answer is option number B, bacterial artificial chromosome. Okay, so these bacterial artificial chromosome can be used as a vector. Okay, these were used in Human Genome Project as well. Okay, bacterial artificial chromosomes and yeast artificial chromosomes were used as a vector for cloning the DNA fragments. Now, why these are used back and yet? Because a larger fragment can be cloned in these vectors. Okay, large size fragments can be cloned. The next question, the primary goal of Human Genome Project was to. Okay, let's have a look at the options. See, option number A is cure the genetic disease. Option number B, sequence the human DNA. Option number C, study the protein structure. And option number D is develop new drugs. What is the correct answer? Okay, we have got the answer. Option number B is the correct answer. Sequence the human DNA. This is the correct answer. See, main goal was to determine the sequence of the human DNA. If you know the sequence of the human DNA, if you know the nucleotide sequence, then you can say estimate the function of that gene. Okay, you can treat the genetic disorders through gene therapy. Okay, so first aim was to determine the nucleotide sequence of the human DNA. Now next question, satellite DNA refers to what? So options are coding sequences. Are these coding sequences? Next is, are they repetitive sequences? Option number C, they are just gene sequences. And option number 4, they are protein coding regions. What is the correct answer? Okay, so option number B is the correct answer here. These are repetitive sequences. Okay, so Satellite DNA can be of two types, microsatellite and mini satellites. Okay, so satellite DNA, they are a type of repetitive DNA sequences. Means these sequences are being repeated. And these sequences, they don't code for any proteins. So they are non-coding. Okay, but these are very important for DNA fingerprinting. These forms the basis of the DNA fingerprinting, these repetitive DNA sequences. Especially the VNTRs, which are variable number of tandem repeats. Okay, this is clear? So with this, we end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.